Thanks. Good to be here. This will be a very good, different talk. I really enjoyed Zubin's talk, sort of laying the foundations, and I'll just be sort of putting to work different sort of statistical tools. So I've always worked in brain imaging since even before I started grad school. I was actually working as a programmer in a PET lab in a hospital, and so I've always worked in this one area of fitting statistical models, building statistical models for brain image data. So I'll just talk about a couple areas of work, but first give you some idea of what I do. I guess I can just do that. Yeah. Oh, it's a little bit slow. Okay, so you've probably seen images like this in the media. People love brain imaging because it generates these really cool images. Scientists have actually done, psychologists have done studies where they've shown people sort of scientific articles that have had or have not had images with colorful pictures, and they believe more the studies that have pictures with colorful, candy-colored brain blotches on them. So the question is, what is this? Why am I interested, and why are people interested? Why does this garner so much attention and actually, frankly, a large volume of research funding? And the reason is, of course, the brain is the seat of who we are, emotion, behavior, and we, relative to other parts of the body and sort of understanding of mechanisms, we don't really understand it that well. And just if you pick out mental disorders, there's a huge burden to society in terms of taking care of people and treating it, and loss of productivity. And we really don't have a lot of advanced knowledge there. And so there's a lot of hope that learning about how the brain works through these brain imaging methods will help us a lot. So also, it's important to realize that these methods are relatively new. Only in, say, the 70s do we start getting computed tomography, and then more recently MRI. And before that, we basically had to wait for accidents. People maybe have heard of Phineas Gage. He was the famous miner who, building a railroad, had a pole blown through his head and suffered a quick recovery, but a dramatic change in his personality, which helped us learn about his, helped us learn where that the front of the brain is where the seat of personality is. Now, we don't have to wait for these accidents. We can just study undergraduates and figure out how the brain works. So the tool that I use the most is magnetic resonance imaging, and I really like it because basically all you have to do is wait, and after a couple of years they'll figure out a new way to generate a new type of data for MRI. So from the same scanner, the same individual, you can obtain all these different types of images. These are just different types of weightings. Basically there are two knobs, something called the echo time and the repetition time, which allow you to have different types of contrast to different types of brain tissue, whether there's the proportion of water in the brain region. You can actually learn about the direction that the wires between brain regions travel. That's this is the anisotropy map. It's really cool. There's lots and lots of different facets of the brain that you can learn about with MRI, and they're sort of advancing every day. And also the images become more and more detailed as time goes by, as they figure out how to make better scanners. So I will focus on functional magnetic resonance imaging, and this is this technique that's actually made possible by this amazing coincidence that we know that blood changes from blue to red as it's exposed to oxygen, but it turns out the magnetic properties of blood change as well. So basically you have this endogenous contrast agent running around in your blood that lets us use a regular old MRI without injecting anything into you to learn about basically how much oxygenated blood is in different parts of the brain. And because the blood supply is very locally controlled, when we use one part of the brain more, there's actually an excess of oxygenated blood there, and we can pick that up with an MRI scanner. So in a very simple experiment, if you just alternated states between rest and then tapping your fingers, in general, nothing changes in the brain because that's a very simple task. But when you find the part of the brain that's responsible for moving your fingers and feeling your thumb touching your fingers, you will find a pattern of activity like that. Psychologists actually never do an experiment. This is too boring. They actually do things that more look like this. They have multiple different conditions, and we have to end up fitting linear models, basically time series regression models, to fit this data, and then they're interested in contrast differences between these different experimental states. I have definitely worked on more complicated models, but it's important to realize that the most sort of the workhorse model is what we call the mass univariate model. So we basically are, even though this is basically four-dimensional data, we get a time image of time series for each subject. we generally use a univariate model fit at each point in the brain as our sort of basis of inference. But there's definitely more fun things to do with that, but that's kind of the workhorse model, which gives rise to an image of test statistics, which then gives us lots of multiple testing problems, which Zubin alluded to. So I didn't want to try to cram too much in. I'll just talk about two different areas that I've worked in. One is in networks, but it's both a small-scale version and a large-scale version. And the sort of the area I'm interested in and hoping that I'll be collaborating with some people through the ATI on what I call population neuroimaging, which is making use of these new large-scale databases that are coming online. So this is work I've done with Jim Smith at Warwick, who's also a faculty fellow here on directed dynamic graphical models for resting state. So strangely, about 20 years ago, someone had the idea of putting someone in the scanner and telling them to do nothing. Usually they actually have them engage in a task, rest, and then remember a list of words, rest, look at these pictures, what have you. And someone said, why don't we just stick the person in the scanner and do nothing? And now the activity is just going to be mean 0, but let's look at the connectivity, the correlations between different brain networks. And this is almost, I would say, taken over task at fMRI as an area of study and interest as people are trying to understand how are different brain regions connected to one another. So with skipping lots of details, basically we define spatial regions of interest in some way. We then get a time series of brain activity for each one of those regions, and then we go about trying to study the dependence between these regions. This is one shorthand way of showing all the connectivity. Here is the full correlation, and here's the partial correlation. And then we're trying to learn about this structure. So we're trying to learn about how do different parts of the brain relate to each other. And so Jim suggests that we look at a multi-regression dynamic model, which was initially worked on by Jeff Harrison, the founding department at Warwick, and Mike West. So let's start with a regular vanilla structural equation model, which assumes independent data over time or across subjects or whatever your measurements are, and we have 3 nodes. So in such a simple setting, we have some sort of independent noise that's going into each node, but then we also have weights. Now, of course, in the standard model, we assume those weights are fixed. In a multi-regression dynamic model, we now allow these weights to vary over time. They, of course, have to be constrained in some way. And what's more, we also are allowing basically a time-varying intercept. So there's white noise, and there's also slowly varying drift that's being added to each node. And so this looks like a much more complicated model, but the thing to point out is that there is not dependence. This node here does not depend on the past. This node depends on the current data. So it's being driven by instantaneous correlations, even though the values of the weights of those connections may depend on the past. It's sort of indirect, and it actually simplifies the model. And without going into the gory details, about this, you actually get a really simple form. The data at each node is a linear combination of the data at the other nodes at the current time, plus some white noise, and then these system variables are constrained to evolve slowly over time. And the really magical thing is that we just need to, if we condition on one, that's on the next slide, isn't it? Skip that slide. If we condition on one factor, basically the variance of the state variables relative to the observation variable, if we condition on that, we actually get closed form posteriors. So conditional on this thing called a discount factor, everything is closed form. We get Bayes factors. There's nothing iterative. There's nothing. It's quite magical. You can do model comparisons, and you can actually easily search the graphical model space, and we can get posteriors on the entire time series of these weights. Make predictions if we need to. This is fMRI. We don't really need to predict the future once we turn the scanner off. We could, but this is the really cool thing that the log model evidence factors both over nodes and time. So you get a really simple expression, and it's very, very easy to score these models and compare them. And then just one last thing is that when I first presented this to my neuroscience colleagues, they said, well, this is for DAGs, right? And it is for DAGs. And they said, well, that's absolutely silly. The brain is not a DAG. Basically, every brain region is wired to another brain region. So there's reciprocal connections everywhere. And so Jim proposed that actually, well, we can basically do kind of what you might say, analogous to the mass univariate model, we could actually have a mass multivariate model. So basically what we can use this framework, but instead of enforcing the DAG constraint, just forget it. We're going to optimize at each node, I'm going to find the best parents for me, and then I'll go to the next node, I'm going to find the best parents for me, and that will not necessarily correspond to a proper statistical model, but it is one that actually can be more interpretable from a neuroscience framework. So we call that a dynamic graphical model. It's basically, well, there's further work to embed this in a more formal statistical framework, but it really seemed to fit the data much better. So I'll just show you a quick application where we have 32 subjects, 15 minutes worth of data on each subject. And now again, this is not a large scale. I cannot apply this to hundreds of nodes, but we've cranked it up to 12 or 15 regions. And these are just 3 of them illustrated here. And we want to try to learn what is the relationship between these 12 regions. If I compare partial correlation on the right, well, of course, partial correlation, which is kind of the interesting thing, which is the unique contribution of one given area to another, it has to be symmetric, right? Whereas here in the DGM, we actually are getting asymmetric. And so here are the parents here, and this is showing here that this important area, the ventral medial prefrontal cortex, has nothing is feeding into it, and it is the parent of a number of nodes here. So this is actually, so here, does VMPC have any parents? No, it doesn't. And when we talk to our neuroscience collaborators, so here's that same network, same image in roughly the same way. They were very excited because this is actually corresponds to the existing models that we have, or existing understanding of the brain, that these frontal brain regions basically are influencing the more posterior brain regions here. And so this is, again, the problem here is we don't have any truth, but it's rewarding that the data that the models that we estimated for this corresponds to what the neuroscientists expected. And I should go back and just say that this image here, we do a Bayesian model fitting for each subject individually, but this is just an image of kind of the consistency across subjects. And so these deep purple is saying nearly all subjects had these connections here. And so there's less consistency, but it's still pretty good. And we actually used a distinct classical method to decide what was consistent enough. But it helped us make sense of all these different models. So that's one example where I've worked with methods for basically doing inference on small-scale networks. But to be honest, people in brain imaging and neuroscience are interested more looking not just at a handful of nodes, but actually the brain as a whole, and basically looking at everything. So the methods I just was talking about is when we basically can carefully pick out the nodes we think are interesting and try to understand the directed relationships between them. People are actually more interested, I would say, or maybe say equally interested, in basically doing more on data-driven view, where we basically put a node everywhere in the brain. We might have thousands or hundreds of thousands of nodes, and then try to understand the behavior of these networks. And we might, for simplicity, and I will in this work, work with a case where we've taken the relationship, the strength of relationship between each pair of nodes and binarized it. So we have a binary connection matrix. So when I first started playing around in this area, I stumbled upon this work from the French group, which was a type of stochastic block model from Daudin's and Robin's group. And basically it was a computationally efficient way to say, what if I'm looking for some latent structure and how these brain regions are organized. I want to find the nodes that exhibit similar behavior both within themselves but across to different sets of nodes. So this is a practical model that because oftentimes these methods you have to assume that you know the number of partitions, the number of latent classes, and this approach actually works that out from a variational approximation. And so we wanted to see, is there some way we could actually test this with real neuroscience data where we know the answer, because in human data we don't. And working with Ed Bullmore and Cambridge, he said, well, why not go to the worm? Because the worm is one place where you actually have a binary data, where you actually say there is a connection from this neuron to that neuron, or there isn't. So there's no fussing about how do you get from continuous data to binary data. And what's more, we know a whole bunch about all these different neurons. We know how they're organized into ganglions, there are different types of neurons, we know what they do, we know where they are in the worm, et cetera, et cetera. And so we can validate the different types of network methods. And in particular, the most sort of if you're trying to find structure, the method that most people use are some version of modularity. And that's generally what is used out there. But I think this was really compelling that when we used a stochastic block model, you see some interesting structure. Now you might say, well, why are these all the same? But actually they are distinguished by how they're connected to other parts of the organism. And then what's more is that you get this, which are our collaborators called hubs, but of course they can't, that's inconsistent with just a simple approach to modularity, which is trying to maximize the amount of connectivity within groups and minimize the connectivity between groups. So this is not sort of news, and certainly, for example, Patrick Wolf and Sophia Olahol have been working on methods like this. But just to say that I'm always skeptical when they first whip out modularity, I'm always skeptical, and certainly biological organisms, I think you really need to be thinking about more general stochastic block models. So the problem was, now I used to be in a biostatistics department, trained as a statistician, but all these methods were developed for single networks. And of course, we live in a world where we have multiple subjects. We want to generalize across subjects, and oftentimes we have things we want to discount, like age, gender. Men's heads are bigger. That doesn't mean that they're smarter or anything, but it does change the data. And so we always, as a routine nature, we always want to discount variation due to nuisance factors. So we want to look at multiple subjects networks, but of course it's going to be difficult if I just ran this algorithm on 3 different subjects, I might get these different networks. There's no reason that they're going to be the same. So working with my graduate student, Dragana Pavlovic, we worked on extending this, so going from the first original model to a model where we consider multiple subjects, but we constrain the latent classes to be the same across subjects, which makes sense. And then crucially, the one that we actually wanted to put to work was basically embedding a logistic regression in that. So we have common network structure across all, but we're allowing, for example, if this is a 20-year-old, 40-year-old, 90-year-old, we're allowing for systematic differences in the connectivity between these as explained by a logistic regression model. So we put that to work, again, with some data from, I got ahead of myself. This is where we assume the effect of the covariates is the same over the network, but we don't have to do that. We can let that effect vary by each class, by each latent group. So again, working with Ed Bullmore, we put this to work on some data that he collected on 25 patients, subjects, their controls, and patients. It's from a larger study, which actually is quite wild and difficult to do. They got to give normal, healthy controls the same drugs that the schizophrenics were on, but we weren't allowed to publish or do anything with the drug side of it. So this is just the control, no drugs, but we have 13 controls and 12 patients, schizophrenic patients. We used a division of the brain into 297 different brain regions. Ed Bullmore has sort of his own way of finding the best way of measuring the strength of association between the different brain regions using a wavelet transform. But in the end, we do just make binary networks out of these. We want to see, can we find patient and control differences? The first question is, are we getting reasonable structure out of this? Are we finding reasonable division of the brains based on these partitions that the stochastic block model finds? And indeed it does. So each color is a different block, and they are kind of arranged anatomically, and these do correspond to known existing networks. For example, this is what's known as the default mode network here, linking areas in the frontal cortex and the posterior cingulate. So that's happy that we got the expected anatomical structure just by estimating what nodes sort of behave like other nodes. And then this is just a visualization. There's no inference yet here. This is just a visualization of the connection, the average connection density across the subjects. So there are 13. This goes up to 13. This is 12 out of the patients and controls. And you can see overall the patients have less connectivity. This is dimmer, but we can include that as sort of a nuisance, or we can test for it directly. And also, of course, we know they have different ages, and so then we fit a logistic regression where at each block we're allowing both for the effect of age and the group difference. And so now we can get an image of test statistics. So these are now based on a walled test. These are the differences between the two patient and control groups. And in general, we see this is negative. The controls have less connectivity, fewer connections, which is what we saw. But we can also look for an age effect, and we also find this is -2. The older that you are, the less connectivity you have. And there's various explanations for that it could be intrinsic properties of the brain. We all actually now happen to know some things about resting state data that people may move to different degrees, and that can corrupt the data to different degrees. So that's an issue that unfortunately on this older data we don't have that controlled for very well. But people nowadays, when they collect resting state data, pay very close attention to the issues of movement. And just as a minor aside, another thing that Ed Wilmore was interested in are these rich block methods. There are a number of graph-based methods that I don't think they're that biologically interpretable, but if the statistical models can enrich them and make them better and maybe more interpretable, why not? So we've taken this rich club method and realized that you can actually use the stochastic block model to create a rich block method, a rich block version of the rich club. And this is just showing in the black solid line is the traditional rich club coefficient, and these colored lines are the approximations you can come up with different sized stochastic block models. And the bottom line is you actually can use a range of different sizes, different number of communities in the stochastic block model, and you're actually reproducing these stochastic, the rich block structure quite well. So that's something I think there's some potential. And I'll just skip this for time. Well, I guess I can, this is just to highlight some work I was doing with Patrick Wolf because the methods that I've been using, even though they're intended for thousands of nodes, they do clog down if you have more than 1,000 nodes. And this was an example of using Patrick's method. We could actually scale this up to 30,000 by 30,000. So really big networks, and apply the same method and detect which of these 30,000 nodes belong to this rich club. All right, in my last 5 and less minutes, I'll just quickly say what's, I think, really interesting me of late are these large-scale projects. Traditionally, unfortunately, brain imaging has been based mainly in psychology departments who study 12 to 15 subjects, and it's kind of difficult to make big conclusions from that. We now have data coming online. This, to me, used to be the biggest study I was ever involved with. This is 1,200 subjects from America who were recruited prospectively the year of their birth. So we know a ton about these subjects. This is a proper population-based study where they have twins. They're selected as being everyone who was born from 2010 to 2015. I'm sorry, everyone born in a certain range in the 90s who was a twin was invited to participate. And it has a target size. There are going to be 1,200 subjects and a huge number of variables that are measured, and state-of-the-art brain imaging data. So I'll just flash some images. The one way we look at this is we can say break up the brain into 50 regions. We can look at the correlations, and then we can rearrange this on the basis of the correlations, and then we can just look at the partial correlations. So I'm going to use the term netmat. Netmat is just the unwound list of all these partial correlations. This seems to be a good summary of the sort of the connectivity, and more interesting than this full correlation here. And so just did one piece of work where we actually use canonical correlation analysis to relate these two different types of things: the brain imaging network data, the netmats, and over 280 behavioral measures to see what connections can we find. And this is with only, I forget the samples, I think it was only with something like 200 or 300 subjects, a subset of the whole sample that's coming out later. And we got this really quite remarkable result. We found exactly one significant canonical correlate, but when you looked at the behavioral variables, so some combination of all the behavioral variables correlates with some imaging mode. And this is pretty sort of stunning. So picture vocabulary, fluid intelligence, delayed discounting, basically how well can you put off a reward to the future, versus do you have THC in your bloodstream right now? And Do you have sleep disturbance, tobacco use? So basically, the first thing that popped out was basically this huge positive, negative life factors. And then we can look at where this relates to in terms of brain imaging modes. And it's a bit more of a complicated story, but we definitely see a strong weight on these, again, this default mode network, these frontal and posterior parietal regions that seem to be important. Now, that was only with 300 so subjects. We've gone back to that with closer to the whole sample, and also in the UK Biobank sample. And unfortunately, it's a much more complicated story. We don't just get one mode. We get, say, 5 or 6 modes with more and more data. So in some ways, we're lucky by the low power to get something simple. The life factors definitely are important, but it's a much more complicated story. All right, so I'll wrap up. There's great interest in connectivity and trying to figure out how different parts of the brain relate to each other. And definitely, I think, hopefully made the point that the modularity has lots of limitations, and there's flexibility that can come from working with stochastic block models. Really interested in the things that can be done with population neuroimaging. There's something called the UK Biobank, which I didn't even mention. That's going to have up to 100,000 individuals scanned with brain image data. Really exciting. They're up to 10,000 already. They've only been at it for a year. And so that data, we have access to that data, and definitely keen to work with people to crack it open and see how the brain relates to everything else. It's up there. Well, the hope is that the other parts of the model are accounting for the temporal dependence. So fMRI data definitely has temporal autocorrelation, and you need to deal with that. But the hope is here that these latent components here are accounting for the slowly varying. And also, there is one latent intercept for each. So yeah, so white noise would be a bad assumption for fMRI data, but white noise plus a slowly varying intercept should hopefully capture the temporal dependence we see.
And when you show this graph with the MPFC at the top, that's like a top-down model. It really only makes sense for resting state data, right? It wouldn't make sense for other type of...
Yeah, I mean, the problem here is that so far we're pushing this model to say up to, say, 20 nodes, but these nodes were handpicked actually for a pain study. So that's why this set of studies, and so this is under resting conditions, no pain, but that's why they're interested in this set of nodes. But you're right, there's other stuff going on in the brain, and that's going to be in there somewhere. But it depends on what you're studying at the time.
In the first part of the talk, is it possible to extract some causal structure by using time delays, or is the temporal resolution not up to?
Very good question. So there has been a lot of criticism, and in fact I've co-authored a paper that criticizes the use of Granger-like causality measures. And the problem is this thing that we measure is secondary, so it's the brain's response, the brain blood flow response to changes in neuronal activity. The changes in neuronal activity are very quick, but the blood response is slow and unfortunately variable. And it in fact can vary across different parts of the brain and across individuals. So you could have a case where you actually have a simultaneous event in the brain in 2 different brain regions, but you'll observe it later in another one. So that's something that I was criticized for this model because there is some dependence on the history here through these weights here, but we've tested it with some simulated data and it seems to be okay, and certainly much less robust to these timing difference than Granger-based methods.
What are the bottlenecks for scaling up without this method?
So the bottlenecks are mainly for this one, and it's just the growth in the number of models. So it is much simpler. I don't know if I have this slides here. The number of DAGs just grows like exponentially, and so there's only number of nodes times 2 to the n minus 1, number of nodes minus 1 models that you have to evaluate, but that still gets big. we make the model simpler by not having to enforce the DAG constraint, but it's still a huge number of models to search. So the things that we've been actually looking at have been doing basically simple greedy searches. I mean, because the standard has been to do something exhaustive, we are looking at, basically, because it's regression in a way at each node, we look at basically stepwise regression methods, looking from the smallest to the biggest model and back again from the biggest down to the smallest to see which ones are, what, as a node, who are my parents, and find the best parents, for a given number find which are the best parents for a given number of parents. And that's actually seemed to work pretty well and gets pretty close to the optimal. So that's one of the ways we've been looking at it.
Any other question? If not, let's thank our speaker.